yesterday. Um, Photosynthesis, big picture, is made of two stages. It's made of a light stage where light is captured. And as a result of that light being captured, we're going to end up with an output of ATP and NADPH. Remember, NADPH is an electron carrier, just like NADH is, and FADH2 is as well. It's got that pH, which does not stand for photosynthesis. It actually stands for phosphate and hydrogen, but it works. If you see on a test it says NADPH, you're talking about photosynthesis. If you only see NADH, no P, you're talking about cell respiration. Okay. So light cycle is the first series of reactions that occur. We're capturing light and we're converting it into a chemical energy. That chemical energy is ATP and NADPH. Now that we have ATP and NADPH, that's energy. Okay? Either ATP directly or the NADPH is an electron carrier, and it can be used to generate more ATP. Okay? Now we want to do something with that. So the something that we're going to do with it is we're going to go into this thing called the Calvin cycle. And in the Calvin cycle, we're going to take all that energy along with carbon dioxide from the air. And we're going to turn that into sugar. We're going to make bigger molecules. So this is an endergonic series of reactions. And we're going to store these high energy electrons in our sugar molecule. And we're going to store our ATP in the sugar molecule. Remember, ATP, not very stable, breaks down quickly. So we have to do something with it before it goes away. And our way of dealing with that is to take that and to put it into a sugar molecule. And that sugar molecule is now our stored energy. And it's a stable version that we can use whenever we need it, or whenever the plant needs it. Okay? So that sets us up for section 10.2, which is today we're just going to talk about light reaction. So light reaction is we're going to take light energy and we're going to convert it into chemical energy. We're going to convert it into ATP and we're going to convert it into NADPH. That's all going to happen in this thylakoid membrane that we talked about yesterday. So we had our stacks of pancakes. And it's going to happen in this membrane here is where this light reaction is going to be taking place. Before we talk about that anymore, though, let's talk a little bit about properties of light. Light is an electromagnetic form of energy, and it travels in waves. It's actually particles traveling in waves. And different light will have a different wavelength. So some of it will have more energy than others. And there's a lot of electromagnetic wavelengths. So we call that the electromagnetic spectrum. All sorts of either high frequency energy or low frequency energy. So what I'm talking about sorry, wrong one. Come on. That versus yoink. Okay, that. So the frequency. So from one crest to another, what's the distance? The longer the distance in between, the less energy there is in the wave. High energy waves are very close together. Right? What we want to focus on for today's discussion is just visible light. And that is the wavelength that photosynthesis uses. And it's also the wavelength that our eyes can process. Okay, our eyes can process some electromagnetic radiation. It can't process everything. It cannot, for example, gamma rays. Gamma rays are really, really high energy, and they're really damaging. We can't see those. Microwaves are very low energy waves. We can't see those as well. We can only see the ones that are in that visible light spectrum, which is shown on the next slide as well. So here I've got, and these things that are up here that we're not going to talk about. So again, these are the ones that are like really, really, really high wavelength. The ones down here are super low. Gamma rays, X-rays, UV rays. Oops. Because their frequency is so high and because they have so much energy, they can actually be very damaging to our body. So although this discussion is not about these waves, they are in general, I like to talk about them because they are important to you as a person, not as a biology student. For example, when you go to the dentist, what do they do when they take X-rays? They put a lead vest over the top, because if they don't have a lead vest, these waves are so powerful that they will penetrate through walls. They'll penetrate through clothing. They'll penetrate through cell membranes. And that's the real problem, because if they're penetrating cell membranes, they can penetrate through the nucleus and to your DNA. And if they penetrate your DNA, they can alter the sequence of the bases of your DNA. And then you get a mutation. That is when your DNA is mutated, right? That can lead to bad things. Cancer in particular. Okay? Once that happens, all sorts of bad things can go on as well. Same thing for UV rays. UV rays are those ones that turn our skin brown or red in some cases. Those waves can also damage your DNA and cause skin cancer. Okay, so we want to stay away from those. Gamma rays, also bad, not as common unless you happen to be an astronaut. A lot of gamma rays in outer space, not so much just hanging out around town. And then below that, we have the infrared, microwave, radio waves. None of these things are harmful to our bodies. They cannot penetrate our cell membranes. They're not going to be able to penetrate our DNA. In this very small range in here, from 380 to 750 nanometers, you won't have to remember those numbers, is the wavelength of light that we can see with our eyes. Okay? And that is our visible light. On the left-hand side, the purple one's going to have a higher energy than the lower ones on the right-hand side, the red ones, the red wavelengths. But this is all energy that we can see with our naked eye. In photosynthesis, most of the visible light spectrum, most of what I showed you on this last slide here, is being used by the plant, except for what wavelength, or what range of wavelength, what color. No, green. Green. Exactly. So when light penetrates the chloroplast and goes to that thylakoid, where those pigments are, those chlorophyll pigments. Chlorophyll pigments are, they trap light energy. They will trap all light energy except for green. Green gets reflected back. And it's the green that bounces back and hits our retina and says, hey, this plant is green. Okay. But it's taking in red, it's taking in orange, it's taking in yellow, it's taking in all those. If you're talking about a chlorophyll molecule. And we talked yesterday about how as those chlorophylls start to degrade, we start to see other colors underneath. But let's just stick with our chlorophyll A and chlorophyll B molecules. These two are both going to be responsible for capturing light energy or processing light energy. And that's when we talk about green and we talk about a plant being green, it's because of those chlorophyll pigments. There is a separate group of accessory pigments that are called carotenoids. And those are our yellows and reds and our oranges. And the same thing would apply for those. They're going to accept the greens, but they're going to reject the yellows or the reds or the oranges. Again, we can't really see those unless the chlorophyll breaks down because there's so much more chlorophyll than there is the carotenoids. We didn't know this for a long time. We didn't know that photosynthesis took in all the light energy except for green until a guy by the name of Engelmann did this experiment that was kind of cool. And see if I can explain this correctly. I'm going to start with the bottom picture here. He took a strand of, what is this? This is Bacterium photometricum. Oh yeah, rolls right off the tongue. This is photosynthetic algae. And so because it's photosynthetic algae, what will it give off as a byproduct of photosynthesis? I heard it. It's going to give off oxygen. Let's go. It's going to give off oxygen. Okay? That's what we're going to breathe. Well, if you're a little, I wish I could, let's see if I can make this bigger here. Zoom in. Maybe. Okay. Maybe not. All right, let's try it a different way then. Let's try it this way. All right. Let me clear that screen off for a second here. Come on. I don't know why it won't clear my screen. We'll do it the old-fashioned way. Okay. So, this is my photosynthetic algae. But then I also, in this medium, Engelmann took, this is in the water, he took a bunch of bacteria that are aerobic bacteria. So to live, they're not performing cell respiration. Okay, they have to get their food from other places. And they also need, if they're doing it aerobically, they need oxygen. They're performing aerobic. That's not saying it here, but they're performing. I'm just going to write C for respiration. Because remember, we need oxygen in order to perform aerobic respiration. So I don't know if you can see this on this picture or not. You see all these little dots here? See if I can make them even bigger. Yep. I'm going to run the risk of getting this so small that I won't be able to do anything else.
That looks like a pile of sand.
I think that's about as good as. There's no way I'm going to be able to change my pointer. Can't read any of that. Okay. So, notice where. Oh boy. Oh, it's gone. Notice where my little dots are located. That's my bacteria. Where are my bacteria located? In terms of color. Blue, purple, red. Where are they really not located? Greenish here, right? So from this and other experiments as well, but he realized that what was happening was this bacteria couldn't survive wherever the light was green, because that was a spot where that algae was not producing any oxygen. And no oxygen was being produced because it was green. So he was able to infer from that that must be why they're not there. They're not congregating in the green light because there's no oxygen. There's no oxygen, they can't live there. They need that oxygen to perform cell respiration. Yeah.
Wait, because the green light's all getting rejected. So
right. And if the green light's getting rejected, there's no photosynthesis taking place. Therefore, no oxygen being produced. And they move to different places. Okay, that's awesome. Now let's see if I can actually. There we go. Let's get that back. Okay, so I talked about the carotenoids. Those are just the accessory pigments that can broaden it. They actually will take in some of the green. So, when you look at a leaf, you do see that there are some carotenoids in them that help with broadening how much of the light spectrum, the visible light spectrum, can be taken in, because they'll take in the green. They won't take in the yellow, the orange, or the reds, but they'll take in the greens. But we're going to mostly be dealing with our chlorophyll A and chlorophyll B. When light comes into a plant, it's going to, that light is going to come and contact the chlorophyll, the pigment. And when it does that, the electrons in the chlorophyll will become excited. What that means is that they get more energy. It's almost like, I'm going to use the tetherball example again. I like myself some tetherball.
I love tetherball.
Isn't it fun?
I really do. Yeah.
Have you ever seen the Gary Larson cartoon, Tether Cat? It's two dogs, and there's a cat on the tether, and if you're a cat person, you're not going to like that joke. So if you're playing tether ball, let's say you're just by yourself, and you hit the ball, what's going to happen to it? It's going to stop? Well, eventually it will, but what will happen to it initially? It's going to spin faster, right? If it's starting to come down, it's getting closer and closer to the pole, and you hit it really hard, it's going to gain energy, and that ball is going to swing around that pole. Okay? That's the same thing that's happening to that electron in the chlorophyll pigment. Light hits it, and that electron becomes excited, and it starts spinning in a higher outer orbital. Fluorescence. or bioluminescence for that matter. But fluorescence here, I've got a Erlenmeyer flask and I've got some stuff. I don't know what it is, but it's changed to this kind of fluorescent color because whatever is in there is photoexcitable. So the electrons in here are changing color. So if you've got like a glow in the dark thing, like a glow in the dark shirt, you know, and you go in the dark and it's like glowing, what's happening is those electrons in that fabric, in that pigment in the fabric, have become excited, and now they're rotating in another orbital, outer orbital, and they're making it glow. Same thing is happening with our electron in our chlorophyll molecule. So let's see how that works exactly. I'm going to skip over these slides and show you the picture first because I think that will be more helpful. Unfortunately, there's no green in this picture, so let's make some green. Actually, I'm going to keep this color. This is my thylakoid membrane. Okay. Embedded in the thylakoid membrane, I have this big old protein. And in that big old protein, I have a whole bunch of green pigments. I have a whole bunch of chlorophyll molecules. I have chlorophyll A and I have chlorophyll B. But I have a lot of them, and that's why I could draw this and make it all green, but then I wouldn't be able to see anything else, so I'm not going to do that. But that's why the plant appears green, because of those chlorophyll molecules. Now, a photon of light comes along, and it's going to yoink, it's going to hit a chlorophyll molecule. And that electron is suddenly a little out of control. It's moving around. It is being passed from one pigment to another, to another, to another. The electron's getting passed. Yeah. Hot potato, hot potato. It's getting transferred from one chlorophyll molecule to another. And it's kind of random in the way it does this. But eventually, it gets passed to chlorophyll A. And when it does that, it gets passed to an electron acceptor who says, like, I'll take him. I know he's crazy right now. I'll take him. I'll deal with this excess energy. Okay. But it takes a while for that to happen. So the light stimulates the electron, and the electron moves around randomly until it finally hits the chlorophyll A molecule that then hands that to the electron acceptor and says, take this. We can't deal with this. Okay. Now, I'm going to come back to these other slides in a second, but I think pictures are better here. There's a few of these that I left out, and I know they're on your slideshow anyway. I do that because I never quite know with College Board if they're going to leave things in or if they're going to change their mind or take stuff out or change their mind. Okay. So, let's make this bigger as well. I want my black color. Let me make sure I got my pen because otherwise I'm not going to be able to do anything. Let's do it one more time. Okay, cool. This purple is it working? This purple blob here is called a photosystem. In my photosystem, I have those chlorophyll molecules that were bouncing the electron from place to place. I have my chlorophyll A molecule, and I have my electron acceptor. Once that electron acceptor says, yeah, I'll take it, I'll deal with this overexcited electron, he's not going to keep it for very long. He's going to pass it to another carrier. Oh, okay. As he does that, a little bit of energy is going to be released to produce an ATP. And he's going to get passed, so my electron's getting passed. And at this point, he's kind of lost a little bit of his energy. He's not as excited as he was. He's like, I'm a little run down right now. But that's okay, because there's another one of these blobs, one of these photosystems. And he will be struck, by another, boom, wave of light. So there'll be a second photon of light that will hit him.
So what's the point of it going from like one?
Okay. So in this first photosystem, we generated ATP. That was the purpose. And as we pass those electrons from one carrier to another, we were able to use that to generate a little ATP. Then it gets hit with a second light beam, light photon. And again, it's going to get picked up by an electron acceptor. He's like, okay, I'll take him, whatever. But this time, rather than making ATP, that electron is eventually going to be passed in a series of steps that you don't need to know about. Oh, that's not what I wanted. Oh my God, how can I erase? Control C? Z. Oh, well, but will it work on that? No. That's really not what I wanted. Oh boy. I have like better in 2020 vision without my glasses. Ah, there we go. Ha! Gotcha. Eraser, that's what I wanted. I wanted to erase that. When you enlarge this, it makes your eraser like uber small. This is the light cycle. This is still the light cycle. Okay, sorry. I wanted to circle this. So the electron eventually gets transferred to NADP to become NADPH. So now I have made ATP and I have made NADPH. And those are the two things that I need from the light cycle to make sugar. Yeah. Yeah. Isn't that fun? Remember when we talked back in the day about oxidative phosphorylation? Remember how I was talking about electrons being transferred and protons being pumped against their concentration gradient, going back down their concentration gradient to generate ATP? That's also happening here. Okay. Electron transport chain and chemiosmosis, that oxidative phosphorylation, that's how we're driving the production of ATP. So once we get those energized electrons, we're doing the whole electron transport oxidative phosphorylation thing. And maybe a bridge too far for today.
Is it the exact same as the other one?
The only thing that's different about it is the direction, the orientation of how the electrons flow in the membrane. Everything else is the same. Yeah. So a great test question that I like to ask, as well as College Board likes to ask, is if there is an electron transport chain in photosynthesis or respiration, or both of them, or neither of them. And the answer is that's both. It's both. Okay. because there is an electron transport chain in photosynthesis that's happening during the light reaction. When those electrons are flowing from carrier to carrier, you're getting that pumping of protons against their concentration gradient. They flow back down their concentration gradient to generate ATP. It's the same process, different system, same process. Do you want the good news or the bad news?
Good news.
Um,
Then
that's not really all of 10.2. The good news is I'm not going to go any farther than that because I want you to absorb this.
I have no idea.
Yeah, that's common. That's
Any